Okay, hey, welcome back to part two for this module. And in this video, I want to talk about a couple different forms of evidence that we use to make some of those decisions when we're making cladograms and constructing and reconstructing evolutionary relationships when doing systematics. So the first one of those is molecular. So, there's a whole subfield of systematics just considered molecular systematics. And this, when doing molecular systematics, researchers are using evidence of like DNA, gene expressions, and whether certain genes are present or absent, or what form they're in, and things like this, and molecular markers and things like that. And there's a lot of different things that can come into play, and a lot of different ways that we can use this evidence. One thing that the molecular evidence allows systematicists to do is sort of extend the scale of phylogenetic work that can be done. So here's sort of an illustration of the hierarchical nature of phylogeny, where you might have a cladogram or some phylogenetic tree showing these evolutionary relationships. You can zoom in. You could construct this pretty much with the fossil record, and you could even zoom in on some of these branches and look at individual species and past species that might have branched off at certain times. But it's hard to really go beyond that, and that's really where the influence of molecular systematics is able to extend this scale. So you can even zoom in within here and look at the genetic pedigree of these organisms and get some genetic information that can really help tease out, whether something diverged and when something diverged, and the evolutionary relationships that might be the result of that and might have preceded that divergence. And so that sort of information basically allows systematics to be applied at the macro scale and at the micro scale, and really extends the amount of information that we can get, and allows us to make much more substantial and robust phylogenetic trees. And going back to what I was talking about in the first video, this is part of what's been fueling this rapid increase in this field of science, is having this increase in molecular data and the ability to use the statistical tools needed to figure out some of this. So this is one of the unique things about it. And just sort of for an example of this, this is a morphological phylogeny of waterfowl and bird species that places this one particular species, 68 to 66 million year old Cretaceous bird, Vegavus. I'm not gonna attempt that, but it's from Western Antarctica, and is placing it within the waterfowl group here. This particular phylogeny has been and continues to be debated about what's exactly going on here. Fossil evidence has some of these shifts in different places, and molecular evidence is now supporting divergence within the Cretaceous period, about 60, within this period. And so this continues to be debated, and things like this get shifted around a lot. And this is something where, sometimes molecular and fossil evidence agree really well with each other. Sometimes they don't. And so having both those lines of evidence can help us sort of sort through some of these things. And sometimes it raises more questions than it answers. But ultimately, the more data we get, usually we can have a better picture of what's going on. The other side of this is obviously the fossil record, and historically within the field of systematics and evolution and those branches of science, the fossil record has been the foundation of evidence that has been used. And so there's a lot more probably evidence that or conclusions and types of hypotheses that have been formed around the fossil record. So we can look at some examples of how the fossil record is used. This is a phylogenetic hypothesis for the family level relationships and the evolutionary history of South American marsupials. And what you can see is down here in the Cretaceous, there was this rapid increase in diversification where you had a common ancestor down here that gave rise to a number of different families here. So just a really proliferation of the diversity in these groups. And basically, I think what's going on here is the solid line represents the known timeline, and the dashed line represents the hypothesized or predicted timeline for how long these groups were around. And there was at one point a bunch of them around, and going up now, there's only 3 of these families of South American marsupials that are still around. But I think the fossil record allows us to make some of these inferences based on, if we just use molecular evidence, we'd probably be stuck with the data that we have available from these here, and maybe you could get some information when these 3 branched off from each other. But the fossil record, we have fossils going all the way back for a lot of these, and allows us to make a much more robust sort of picture, gives us a much more robust picture of the evolutionary relationship. So you might think that like the molecular tools are way more advanced, but we're limited in some ways in how far back we have access. We have that type of data. Fossil records go back much further than we can get molecular data for in a lot of cases. So it can give us a much more complete picture. Not to say that they don't complement one another, but fossil record is used very extensively. But there's limitations in the fossil record as well. So here it's kind of, I like this figure. I've never actually seen this figure before until I was putting this together. But this is estimated area and volume of potentially fossil-bearing sedimentary geological strata of varying ages. So basically, how the volume of basically Earth that could contain fossils by area and by volume going back through time. And there's interesting stuff going on in here, but I think the general trend is just that if if we'll start with area, you get a decrease as you go back in time in the area of available soil that might contain, or geologic strata, I shouldn't say soil, of the geological strata that might contain fossils. And so that just decreases over time. And a lot of this has just been lost over time. And so, it would be reasonable to assume that as you go back in time, you're going to find fewer and fewer fossils because the material that contains the fossils decreases, the amount of it decreases as you go back over time. Volume, the relationship isn't quite as strong, but I think it's still there. Most of it's in the Cenozoic, and then as you go back in time, you get a decrease in volume as well. So basically what these result in is fewer and fewer fossils from older times, which which is what you would expect. The older it's been around, it has more time to just decay and weathering and all those sorts of things that occur to the surface of the Earth take place, and so those specimens just aren't as prevalent from far back in time. So we're limited in that way too when you're using fossil type evidence. This is an interesting one. This is a morphological. You're looking at the morphological characteristics of one of the earliest known bats, which provides strong evidence that flight had evolved at least 52 million years ago, which I don't remember exactly where the estimate was before this, but this is an interesting. I'm pretty sure when this fossil pushed back that date quite a bit to 52.5 million years ago. And you can see some of the sort of morphological characteristics that contributed to that. So elongated digits and things like that. And they even had some rudimentary structures that allowed for sort of echolocation that bats are known for. This fossil, I think the estimates, looking at this, they don't think that bats of this time period were able to use echolocation to the extent that some species are now. but was some early versions of those characteristics. So an interesting fossil specimen there. Here, these are fossil localities of the camelids. So I think the camelids came into existence around 40 million years ago, and they were really widespread, existing all the way up into Alaska up here, and all the way down into Central, and eventually got down into South America. So they're really widespread all the way up until about 20,000 years ago. And there's this really interesting book, which I think I have a couple figures somewhere in this module from that, but it's called The End of the Megafauna. And it talks about, a bunch of species that existed around the world that all went extinct around the same time. And we actually talked about some of this when we talked about the Pleistocene, and talked about how when a lot of these species came into contact with humans, they tended to go extinct. That's probably a big part of the story here, but maybe not all of the story. There's climate stuff that was going on, and there's other stuff going on, and so it's an interesting story. But there were all these camels and species in different genera too, and there are lots of fossils, really widespread, all the way up until about 20,000 years ago, where they abruptly kind of disappeared. And they found fossils, I think frozen fossils, all the way up here in Alaska, that helped tease out some of those genetic relationships. But there's still some in South America. It's not on the map here, but in South America, some of these species and descendants persisted. So interesting story there. This is the lungfish order Lepid. I'm not going to try. I'm not going to try to pronounce it. But it's represented here by 5 species on 2 of the southern continents. And basically, what you're seeing here is, so you have this out group, which is represented. There was this whole group of them, and that's what you're seeing in the red dots. Those are fossils of this group here. But now they're only represented by 1 species. Which is this one here in Australia. But there's these descendant clades that you're seeing in the green here in their distribution. So basically they were really widespread, really widely distributed around the world, and they have this really restricted geographic range now. So it's interesting. And molecular evidence has provided some additional information about when these might have split off from one another in terms of time, as well as their evolutionary relationships to one another. So we can sort of combine those and get some additional information, and then we can use sort of biogeographical stuff and look at, plate tectonics and what we know about where where the continents were, and where maybe coastlines were different, and maybe continental seas were present in some places that no longer are. Specifically, we can look at things like the global distribution of limestone, which we know forms from like coral and marine aquatic environments. And we can see this distributed, this is something that we have here in Bloomington. We have a lot of limestone, and we can look at that and say, okay, if we know the certain types of environments that produce these limestone, then we can say, oh, there must have been marine type environments in some of these locations at some point. We talked about the intercontinental sea that basically bifurcated part of North America at one point, and so we can use information like that to get some additional biogeographical context for these fossils that we find. This is an interesting one. So this is a geographic range shift from the mitochondrial DNA used to phylogenetically reconstruct a group of related leaf-eared mouse or leaf-eared mice. These guys here. And there's a bunch of different species of these in South America, located here. They found a feces sample here that was like almost 12,000 years old. I think it was 11,700, if I remember right. They found this sample and were able to reconstruct some versions of the genetic sequences. And basically they put this into their phylogenetic analysis and did some statistical analysis and said, where does that fall? If you include this, where does it fall in similarity to these other species that exist now? And when they did that, it was this P. lamatis species that was most closely related. So they basically assumed it was that species that was here. And from that, we can say, okay, we found this sample here. That means that P. lamatis range must have extended down here, probably, probability wise, it was probably extended further down here if we found this sample. And so from that, we can sort of infer like, oh, there's been this range shift since 12,000 years ago when they found the sample. So you can draw some interesting biogeographic inferences and conclusions from some of this stuff too. So that's kind of interesting. Okay, so I'm going to wrap up this video. There's a reading here. It's a little bit different. It has to do with fossils, but it's this interesting story about a scientist named Mary Anning, who is a fossil hunter who until recently did not get the credit that she deserved for her role in collecting tons and tons of fossils in England around the shores there that contributed to a lot of scientific breakthroughs. And so this is a story of a lot of science that people in the past, if you're a woman or a person of color or anything that isn't like a straight white wealthy male, you probably were overlooked. And so I wanted to include this because I thought it was a cool little story. So read through that, keep working your way through the module, and I'll see you in the next video.